I don't know. Heading well.
Yeah. Curious about auto XY. Auto depth.
I think the question is whether you'll move with auto XY.
So we had a really good question here in the control room, and that was that it seems like sort of the middle of the spires that we're looking at is actually thinner than the top piece, so it looks like it's a little top heavy. It's got some flanges on it. It's got some sort of broadening up as opposed to narrowing up. And the question was, why is that so? And there are a number of different reasons for it, but a lot of it is that basically some of the fluid can sometimes sort of bust out of a side, and it will then start creating new mineralized areas. And then the other question that went along with that is, do these ever fall over?
So we're going to let this, see how this goes.
Absolutely, yes.
You can come into zoom, but when I say
active area,
come out, you need to come out in a hurry because I might be getting pushed around.
But
right now it's holding very steady, so I'm trusting it.
Over and over again. There have been ones that we've gone back to and said, oh, what happened to you?
If you need me to get out quicker
and what's
than I
often
think you do.
about when these vents fall over,
I'll try to keep you informed of what I'm doing as far as this
growth over time. And you often can see the changing mineralogy between, sort of the young vent and how, as it gets older, how it mineralizes. Oh, look at that orifice.
Good job.
Look at those shrimp. Oh, I'm sorry, supposed to be looking at the shrimp.
Supposed to be looking at it all.
You can actually see here how the fluid is coming out actually clear.
Yeah.
And then
it immediately
hits,
changes.
Yeah, that cold water, those particulates begin to participate out of the
going to
the
edge of the chimney.
And so here you can see these shrimp are probably in water that's about 20 degrees Celsius at most, and you can see the white patch on the the dorsal surface of their head is. Those are sort of, they're not eyes, but they're modified retinal systems where they're able to pick up
commands in auto XY.
Yes, no, I know. I know. Yeah. I don't know who's sending it.
A very small amount of light, which is emitted from the actual orifice of the chimney.
I'm not going to do anything but change heading and
so that is likely what allows them to, apart from being able to tell the difference in temperature, that's one of the things that helps them to
Thank you for the reminder.
Where they need to get to get as close as they can to that vent fluid, where they can bathe themselves and make that vent bacteria, which they need to survive.
Now, are these the same shrimp that we saw at those diffuse vents a couple of days ago?
Very likely so. And so
it's nice piloting.
Many of you may have been tuning in. About 3 days ago, we visited an area of diffuse flow.
Awesome.
So it looked, the water just looked very shimmery like this. There wasn't any black fluid. And there we saw 3 to 5 shrimp just sort of hanging around in that diffuse flow, and that's all they really need in some cases to survive.
Right. Stace is saying that this morphology is consistent with Chococaris. Is that how am I
so this is Chorocaris.
Chorocaris, sorry.
We're not sure which. There are 2 species here.
And you can
see one of the younger ones that's just going under the rock. So, hold on. Okay, I'll try to find another one again.
What is different about the younger ones versus
they're smaller, and then they're also slightly redder in some places.
Okay.
Yeah, trying to get there carefully with
I just wanted to let you know where I was going.
Auto heading.
So, I mean, you can see all the diffuse flow. You can see, well, now you can see the black smoke as well. But you can really see that these are oases of life, exactly as you were talking about, Diva. Yep. These are areas where the density and abundance of life is very, very high.
Going to go tight to the shrimp level at the
fairly
bottom.
Sparse. Yeah. And so it's really just amazing when you have a chance to look at this kind of stuff. I don't know that I've ever seen these kind of swarming shrimp things before. Because
have you worked in the Atlantic?
No, that's exactly
so this
it.
is
like
nothing on the Atlantic.
I've seen pictures of this.
Millions and millions. It literally looks like a swarm of animals just constantly jostling to try and get close to the fluid.
So really, I have an important question.
So that's a young one at the bottom of the screen.
Oh, the little redder one?
Yeah.
So I have a very important question, Diva.
Yep.
How would these guys taste?
Pulling out to find the
Probably
chimney.
not very good because they have sulfides
just rotate for the chimney.
When you bring them up, they smell a little bit.
No,
and they're also only about
look at that orifice.
6-7 centimeters across.
The one that are behind the other flows, I can't get those in focus due to the
building their little chimneys, tiny, tiny little chimneys.
Okay.
But behind, so there's a chimney in the foreground,
right?
But in the background, it's almost like beehive venting, where it's coming out very slowly.
Yep.
Oh, look at the beautiful beehive on top of that
Oh,
one.
that is gorgeous.
Oh my God,
Can you zoom
amazing.
in on that beehive?
Which is
It's to the left of the black
top
smoker.
left one.
You see
okay.
that it looks
Yeah.
like a little
Continue
pine
at this
cone?
speed.
Yep. We'll tilt up.
So,
Rotate a
shore-based
little more.
scientists, I think you're going to have to break in and stop us from talking every so often, because I think
Start
otherwise
in.
we're going to continue to narrate this. But I'm sure there's really good conversation that's
Tilt it a little
happening
more.
at our
On
exploration
shore.
command
Yeah.
centers in the event
And
log.
rotate it
So
left.
please feel free to
There you
break
go.
in and tell us what you're seeing.
brought you
tell
in at
us
10
why
meters.
it's exciting.
Thanks a lot. Yeah,
Please
yeah.
let viewers at home know why
Turning my
we
head, I want to make
are
sure
so
I'm
excited
not getting
about
too
this.
close to anything.
Why are there only shrimp? Shouldn't there be something else?
There are on the... Oh, sorry, Diva, go.
So that's because at vents you tend to get a very classic zonation.
Can I push in
and that depends
co-pilot on Sirius?
on the concentrations
Yeah, I
of
think
vent
so.
fluid and temperatures
We're
of vent
just
fluid
now going through Hughes,
emanating
actually.
from various
If you wanna-
regions of the vents. So in the most high temperature areas, you tend to get
Okay, we'll
sort
go
of
pull out
shrimp
ROV.
and, well, it depends which biogeographic province you're in, but we thought we'd be seeing some tube worms here, not the riftia, but the, is it
That's-
Paravanella, I thought? But you tend to get shrimp and crabs in, and also some gastropods in the areas of most high venting. And then as you move away from the fluid, animals tend to get sparser.
What about the
Okay,
polycule worms? Where
pilots,
do they
I've
tend to
imaged
hang
So
that
out?
they're
area.
sort
If
of
you want to
midway,
look at anything
mid-region.
else,
And then as you
Okay.
move
I
into the
eventually will
peripheral
want to look
regions,
for
you
a
tend to see the
crab that we
anemones
haven't
sort
seen
of...
yet in
So
the
it's,
tights.
they are very classic zones.
Oh, one of the crabs.
Yep.
Okay.
Pilots, I have a question.
Go ahead.
Would there be a good time to deploy the high T probe at some point in time?
After we're done
Would you
imaging.
feel comfortable?
Yeah, once we're done imaging, if for some reason we bump something, I hate for it to not get a good shot of it before we do bump something.
Perfect. Okay, so shore-based scientists, what we're going to do is we're going to continue to image. We're going to get some really nice
if that's
zooms.
okay with you guys.
And once we've had a chance to do all that, then we'll deploy the
Okay,
high
if you want
temperature
to,
probe.
just move to whatever area.
Because we want to make sure that, you know, as we all
Yeah,
know,
I see a crab down
sometimes
low. Do you like that
probe
one?
can
Yeah, I don't see it yet.
do
but
a tiny
okay.
bit of damage
Yes.
to the chimneys. Just a tiny bit.
Well, yeah, you're sticking a temperature probe in and near it,
Using
so
a high temperature probe
we'll try not
on
to
the flange of
damage
the
anything,
bottom
but
because
you might chip the
those usually have
end
very
off or
hot
something.
fluid under
It is
the
a floating
flange, and
vehicle
it's
with a
relatively
probe
non-destructive
sticking off of
way
it,
to measure the
so.
temperature.
That sounds great.
200
Okay,
meters
yeah, I
down.
see bottom right of the
Into the
lasers. I see two crabs.
Yep, lining up on
That's
that.
a really great
Oh,
point,
wait.
Bill. And
Zoom
so
in when you're ready.
actually, when we start getting back into the area where we see the
Come on
flanges,
in.
just shout it out so that we remember.
Okay, there's one right at the very bottom. So
Yeah, I thought it was one of the first things we saw. Yep, thanks.
It's hard to focus through all the flow, huh?
Yep. Oh.
Oh, look at these guys.
Oh, there it is.
Oh, sort of look at these guys. There we go.
So
Crab?
these are bifidograde crabs. They're likely from... Oh,
I like the way the shrimp are just sort of moving in on the crabs. Like, hey, get out of
Tilt
my way.
up. Maybe we can get two.
Trying to get over here.
oh,
there's
look
the
at that
bottom of one.
He's sunning himself.
Yeah, not imaging them great.
Okay, well we
due
can,
to the
if
flow.
we see one without flow, we'll
Yep.
take a closer look at them. It's hard to shoot through the flow, I understand. Alright, I'm going to
One
come
down below.
Ooh, there is one down below, isn't there?
And we can also see the orange
I'll come out of auto
full wide.
here and fly to that guy down below.
This could be
So stable. It's
Co-pilot, you let me know when I need to pull Sirius further out. It's a great image right now.
I think we're good here for now.
I'm going to drop
Will do.
down and move over. There's a few crabs over here, so
At some of these vents.
You dropped a target right now?
There might even be a possibility of brine coming out because it's separating red to C4. So
Bob, could you explain this
We
a
have a
little
target
more?
on this,
Yeah.
yes.
Could you
Yeah.
explain phase separation a
I
little
named
bit
it
more
waypoint 1
and
adjusted,
why we might expect to see brine?
and then I dropped a waypoint 2 adjusted
Initial zoom.
on the assumption that we are on waypoint 1.
Okeanos.
Yes.
He's about to go behind.
Yep, I know, but
Call him out.
like 400 degrees.
I want the autos to settle before we do any zoom so I can be comfortable.
So you might be seeing phase separation
Yeah, see,
pattern
they're just going to wander away like that,
C4.
possibly.
So there might be brine coming up.
Oh, there's one right there, though. Hold on.
Thanks a lot, Bob.
Come in a bit. See the guy?
Tilt down.
I'm going to tilt up.
Oh, I was looking for the guy that was on the orange below.
I think he's not in very much flow, or he wasn't. Now he's in more. Okay, well, you can keep it here. I'll tilt the camera and change the heading and see if we can.
The guy on the right
and
edge
so
lower
Stace, to
of
be
the
able to
screen
get that, we
is
need to stick our HD
closest
probe
to
in a
the
very black
non-flow.
smoker, correct?
No, not necessarily. Did you hear what Bill was saying?
No.
That there's actually often very high temperature underneath the flanges.
Right. Okay. That I know that is where one of the sharpest
Which one would you like?
gradients is.
Up or down?
Temperature gradients is.
I think the upper
So this
one.
bifurcated crab is likely
big
the
claw on that
species
guy, huh?
Ostinagreo williamsi, which is known from this area. And these are just absolutely characteristic
best
of
image.
Pacific vent fauna.
He hid from us,
All right.
pulling full wide.
Yeah. Well, you can keep it there. We'll just scan around at this.
Have I said this is amazing?
Totally
Yeah.
amazing.
See if we catch
And
another one
again, for
crawling
those of
around.
you who are following along at home,
They are chasing moving targets.
what was the question?
Looks like that guy at the bottom of that one, just above your lasers. Maybe a good
Okay.
character now.
Yeah, come in.
Pushing in.
Get him before he gets behind there.
So these animals, despite the fact that they're living, what you think
Yep.
they're living in very high temperatures, they're actually only living in about waters between sort of 5 and 25 degrees Celsius usually. And they can get burnt, just like you and I would if we were to put our hands close to this. Occasionally when you bring up vent fauna, shrimp, crabs and so on, you do see that they've had a couple limbs singed off when they do end up getting too close to the fluid.
Ouch. Ooh, try this
So
guy.
they need to put themselves sort of in that perfect area. Sorry, what was that, Bill? Or Bob?
I mean, this whole thing's covered in
Bob. Sorry, Bob.
in
What was
flows,
that?
I guess. It's hard to see through it. Did we already try this one up at the top? Come in again.
Oh, you are not coming in clearly.
Come in partial.
We can't hear you at all, so if you want to type it into the event log, please do.
Yeah, he's behind.
Partial.
Flow.
I
Thanks.
think you're gonna get flow everywhere.
Yeah. We'll see, like, these pokes
So,
here
Bob,
where
we
there
couldn't
isn't.
hear you, so if you
Get
want to
that one
type
up
into
there.
the event log, we'll be
Just
able
tilt
to
up
find
to
it
those
there.
two.
There's a crab facing us just upper right of lasers.
Yeah, we're headed towards it for you. Hopefully there's not too much flow between us and him. Coming over. We're going to get some, you know, I think
Yep.
everywhere. So we'll take the best we can. You can start in. Come in on the crab facing us.
Pushing in.
Go as tight as you like. Vehicle steady.
I think that's a pretty good view, pilot. I think that'll give the scientists the closest description of them.
Okay, I'm going to tilt up real quick, stay at the zoom. There's another one back in there. I don't know if that's a good shot too or not. I guess not really. But get a good view of the legs and the carapace. Okay,
Which is a 10-story building.
so we'll try for the little dots next.
So
I
Bob
think they're
may
up
have
from
said
here.
this already, but this chimney is
Following
30
you, I
meters
can pull
high.
out as
That's
needed.
essentially
Yeah,
a
let's pull out.
10-story
I'm going to
building.
have to fly over there
Okay.
and
Pulling
catch it.
full wide.
And those guys are still, so hopefully you can pick a real good one and stay, or they're stiller. So if we can pick a real good one and stick with it. Out of autos. It's so nice that the autos are holding well.
Yeah, definitely.
Makes this job significantly easier.
Devin,
Be a lot
Eva,
more comfortable.
for a little variety, it'd be nice to
Remember that big mess of the
move
little
over to
circles?
one of
Where'd they
those
go?
places that
Are they
had a
up
lot
more
of
from here?
gastropods. We haven't had a
What's that?
look at
Remember
them
that
yet.
mess of the
Okay,
little
great.
circles
we will
that we
ask to see
saw?
if we can move over to the gastropod areas.
Yeah, I think that was way down.
Way down
So,
or way
pilot?
out? Yes.
When you have a chance, could you try to find one of the areas where there were a lot of gastropods?
Ooh, we read your mind. We're headed there right now.
Excellent.
So you can
thank
see one on the right, just where the sort of overhang is.
Yeah. This general depth is where I was headed to, and I was going to
Yeah,
get into
look for
autos
polka
and
dots.
hold here and get a good zoom. Hopefully. It's through the smoke, so we'll do what we can.
Okay, pushing in.
Nope.
Oh, sorry. I thought you were talking to me.
No. Does that look safe to you?
Yeah.
Everything looks okay, right? Yeah. This is what I think is going to be for the best view for these polykeet guys. All right, you can start in, Roland.
I think they're gastropods.
Or gastropods, sorry. I keep calling them polykeets.
So I've just heard Dev explaining in our control van to many members of
you can zoom if you like, Roland,
of how
when you're
these
ready.
are created.
Pushing
So it kind of reminded
pilot.
me that we haven't really done that. So in a nutshell, what tends to happen, and this is the biologist explaining this, is there tend to be fractures in the seafloor, which seawater percolates down into. And once it gets further down into the crust, it basically becomes heated and also changes its chemistry. So it will drop certain minerals and it will pick up certain minerals. And then, once it's heated, it becomes buoyant.
I think this is
and again,
probably the best
moves
section.
up and out of the crust and is
Yeah.
expelled
So
into what is very cold water. The vent fluid is around 300, 400 degrees Celsius usually, and once it hits that cold seawater of about 2 degrees centigrade,
down sort of helps.
it tends to make all of the metals, well, not all of them, a lot of them precipitate out of the vent fluid. And that's why you get this very black, what looks like smoke.
You feel like you get a good shot with this. We can try scanning around all other places to see if it's better.
A lot of the metals precipitate around the orifice that the fluid is emanating out of, and it builds up over time, creating these chimney-like structures that we're looking at.
Straight up is the next best.
And so
I'll scan
what I
slow
think
up.
we can see right now are a type of
like it was less
gastropod
flow.
or limpet. I'm not sure which one. I'm not sure if scientists on shore know. I think Stace has just said she thinks it may be Shinkai-lepus, which I'm guessing is named after the Japanese human-occupied vehicle Shinkai.
See if there's
we
anything
have to
else.
assume that that's exactly why.
Which I think has done quite a bit of work in the Marianas area.
There you go.
So these look like maybe bivalves.
Yeah, some kind of.
Science, are these potentially bivalves we're looking at here?
No, so they're
gastropods.
limpets.
So that's like a single shell, isn't it?
Yeah.
Or
Exactly.
oh, I can't even begin to tell you how happy I am right now.
Okay, pilot.
Diva?
I think I've got the best I can get of this.
Yeah. I mean,
Okay.
yeah, this is everything we've been
Going
waiting
out.
for
Yeah,
for the
pretty
last
much.
like 10 dives. Not that they've not been great.
Right, and not that we won't be excited by the rest of our dives
exactly.
here on this
But
expedition.
this is
It's a
kind
whole
of
ton of them. So
both
this
see
is
us
what
a
I
lot.
do.
Oh,
being
look at that.
Actually,
hydrothermal
I
vent
think that
people
section in
tend
the red
to
zone left of your
favor.
lasers.
And I think we can say that about
And the red
many,
left of the
but not
lasers.
all people on
Is-
planet as
actually,
well.
maybe
so
right
in
where you're at,
addition
we
to
can
the
check that
hydrothermal
upper
vent
area.
scientists that have
Okay.
come out
Go ahead.
to help us
Okay.
and help explain what we're doing and how we're doing it on this particular dive, we still have a lot of our
Wow.
I don't want to say
That's
regular people, but we have a lot of people that are
Close
just
shot.
interested in exploring the seafloor, who are interested in
Look
corals
at
and
the structure.
cyanoids
Look at
and
this.
any of the other
The thing that it's
biology
on.
that are not necessarily found in
Look, the
vents.
smoke just came in.
And I
Yeah.
think this is an
awesome.
appropriate time
Yeah.
to also point out the all-star team
I
of
should say
hydrothermal
minerals.
scientists that we have on shore. I think, as Deb was sort of saying
Touched
yesterday,
left a little,
in
see if that
combined
centers it up.
experience at hydrothermal vents, we've probably covered all biogeographic
But
provinces.
all things considered, the autos are
amount
doing
to
a
over
really,
100
really, really good
cruises,
job.
well over 100 cruises,
We are
vent
max zoom
cruises
right
between
now.
us all.
We really
And
need to start adding
we-
these.
We are keeping
Bill,
a
how
tiny
many cruises have you
gastropod
been on, do you
almost
know?
in the center of view.
Yep.
Really
Hey,
lucky.
Donnie.
Something that Dave Wright used to use is actually on your control screen where you bump left and right.
Yeah. It's hard. You got to be able to judge
Pulling out
how
full
many.
line.
How many? I think it's better to fly and resettle each time. It seems to be working nicely.
Agreed.
But I do know what you're talking about, and that is a good suggestion, Roland. I fear using it. So,
I don't think this is an appropriate place to use it. Really.
You guys know
We
it.
have not talked about sampling
It's
yet,
really-
but before we talk
Oh,
about
there's a
sampling,
great
Tim, I
crab
think
at
we
the
have to talk
top
about the fact
left
that
of
you
the
are on
lasers, not
another
in the floor.
U.S. research vessel
top left.
watching video. So they're
Oh,
on
I
the
see.
Armstrong, which
He's
is
crawling?
one of, it's actually
Yeah.
the newest
You can start
ship
in.
in the U.S.
Try to get a
academic
view of him
research
before the
fleet.
Probably the
cloud
one
hits
we
him.
get the best.
And,
Becomes
and
a
it
bloom.
sounds like you are watching it from the ship. So that is really, really cool. Now we've got a whole lot of weirdness going on. I like that. But now I will let Diva talk about the real important question that you asked, which is sampling.
Sorry, so Tim, before on the pre-dive call, a couple of scientists did chat about sampling. And
He went straight into
there
the
were
flow.
some people raised various animals,
Slow-
species,
slow
which they thought
methodical
they
moves
wanted,
for
but
us, and these guys
we
are
kind
quick,
of just
so.
reiterated that
Sorry.
The Okeanos sort of philosophy of sampling is that we like it to be novel and sort of help to characterize the area essentially overall. So as much as we want to help with, for instance, connectivity studies,
I said
that's
if you want
not
to look
really
at
tying in
another
with some of the goals that we have on board. So I don't know, do you want to chime in with anything
Let's see what
that
scientists maybe have another
would be particularly
thing they want to look at.
sought after?
Any other zooms?
Well, I don't.
Maybe a little tilt down out of the
in the
flares.
Mariana region,
Copy.
and on seamounts that are hydrothermally active and have experienced recent eruptions, we see a similar species. We don't know that until we sample them and look at the genetics, but they're similar species. But the Mariana region is specious with shrimp, and so I could not say based on what I saw that
can't hear
the
you.
abdomen
I don't know
is
if
so
you guys are
thick,
talking
I
to
couldn't
us.
tell if they were eggs on females, but
the
the
front
abdomen
row.
is so thick, it's characteristic of what we see in the Atlantic with Coracaris chasei. And so, anyway, it looks sort of different to me. I haven't seen a real, you got great video, but I haven't seen a specific zoom in on a really clear shrimp yet. But anyway, I don't know if it's new or not. It's a thrill to watch this, though. And I'm just curious. You'd have to have a suction sampler to get the shrimp anyway. I don't know if you have that on board.
To see what?
Unfortunately, we don't have a suction sampler.
Another thing that we're all keeping in mind is that we're going to be returning to
this is a whole mess of them if you want to look into it.
With
Try to get
more sampling in
a little look.
then, so
Come on in.
it's not a completely lost opportunity.
Right,
that makes great sense. Yeah.
See if this.
Okay. Thank you very much.
Actually, bottom.
We're going to keep watching. We'll chime in
Thanks,
when we can.
Tim. Keep watching, and if you do see anything absolutely incredible, or you want to chime in at any point, please feel free to do so. And all scientists on shore, the same for you guys.
All right.
I love that we just received a comment in the event log from a geologist who noted the upper portion of the chimney is shrimp land, and the lower portion might be snail land. And I've been looking in particular for the snails in the background, and you'll note very low diversity of snails. I mean, pretty much we only see that one limpet up here in the high temperatures. But I'm excited to check out the base of the chimney or elsewhere and look for some more diversity of snails.
And that's exactly right, Stace. And just as Bob pointed out, you've got shrimp land, and then we're hoping what is going to be snail land. And then in the further peripheral regions, you get the squat lobsters and the anemones than when we were flying in that we noticed. And so it's that very clear zonation that I was talking about earlier.
We also have another scientist tuning in, I believe, from Canada, who's wondering where the barnacle land is.
Have we seen barnacles
No,
yet?
so Santiago, we have not seen any barnacles yet. Relax.
All right. I'm ready to come wide, I
That
think.
suggests slowly moving down the chimney.
Go back down the chimney. Aye. That's the only command I've heard in a while, so I'm going to go for it.
Yeah, I would. Would it come wide from you, Roland?
All right, so we think...
Pulling wide on Sirius.
So I think that the next thing that we're going to do, Bill, I think you just actually said this while Diva and I were talking about it. We're going to move down the chimney a little bit.
Copy.
We're going to try to find those areas that are maybe a little bit more of the mid-temperature zone. But we're also going to be looking for a good flange where we might be able to take a high-temperature probe. But we want to make sure that we take the imagery beforehand just in case anything might fall off during the temperature probe. So I think we're just slowly moving down the chimney a little bit. And Diva, please scream out and tell the pilots when to stop.
And I think we've already said what an incredible
cool.
onshore base of scientists we have joining us today. But I think it's also really important at this point to point out that we have an incredible ROV team on board who's getting this incredible footage. And we've also got an incredible group of crew on this ship who are totally responsible for making this happen. And we really shouldn't forget about them. A lot of them have been in the control van today and are watching, just as intensely as we are right now, just as excited as we are. And I think we should really take this opportunity to sort of mention them and thank them for helping this all to happen.
I absolutely agree. And I mean, this is beyond our ROV team, which is fantastic, and they're doing a really, really great job of getting us imagery, doing sampling, and making sure that we end up in the places we want to end up in. All of the people on this ship are interested in the science mission that goes along with what we're doing today, and that's everybody. And so it's really great to know that they can come on in and talk to us in the control van, and they're really excited to learn about what we're doing.
So excited.
Coming down with it.
Copy that.
So
You muted. I can't. Say
so
again.
we can see that there's a lot more gastropods in this area, and I think the further down we go, the more they'll increase. But is it possible to get a quick snap zoom just to see if these are limpets or snails?
Yeah, come on in,
Please?
Roland. Let's make it a snap zoom just to determine what it is.
Okay, so still limpets.
Still limpids. I agree.
Oh no, we can, okay, so we're beginning to see some gastropods. So you can see, well, they're all gastropods, but beginning to see some classic snails off to the right. The darker shells. You can see they're not sort of flat. They're just the classic Alvinaconcha. Stace, are you seeing this?
That is max zoom.
Thanks.
There are 3 biologists here who are very happy in Woods Hole.
Got everything in this image.
Okay.
Crabs, shrimp, and limpets and
Thanks.
snails.
That's
All
great.
right.
Stace, do you want to point out who they are? Just
come out
just
when
for
you're
viewers,
ready.
just so we know. I mean, I think it's just very interesting that we're able to connect with vent biologists who are all the way in Massachusetts right now.
Okay, pulling out. There's a red shrimp that I want to comment on.
Here in Woods Hole, Massachusetts, I'm Stace Follieu, and
Is that
I
a
have
baby
with
shrimp,
me
Red?
Susan Mills
That's what they said
and
earlier.
Lauren Molyneux,
Cool.
and we study animals that live at deep sea vents. And we also
Actually,
have
current's
Julie
not so bad, I
Huber,
guess. I
a
can hold pretty well.
View on
microbiologist who
Sirius.
studies
Doesn't take a whole lot
the
to,
bacteria and archaea that live at vents.
It's probably
Thanks,
worse the higher
Stace.
you
That's
get.
awesome.
Yeah, for sure.
Stace was just introducing all the scientists at Woods Hole.
Got a great view
That's
on
awesome.
Sirius.
So you can actually
Those
see
little
here
red
the
ones.
2 little red ones, yeah,
And
which
those
are going
are
to be
the-
juveniles.
Those are
I'm
like
thinking.
the adolescent shrimp.
I mean, no, I think they're even less than adolescent.
Oh.
No. Yeah.
They're not yet in the
Maybe,
teenage,
maybe.
in the pouty teenage stage. So what was Tim Shank saying about the abdomen of the shrimp? Did you understand that, Diva?
Oh, I didn't. Hey,
Oh,
sorry.
I just
It's okay, maybe
can
it's
scan
somebody else
around
can
with
read.
a little
What
zoom
was he
here.
saying?
I don't know. All I heard was the
Was it
word
about
Catch anything else
bacteria?
interesting,
shrimp.
but
I have no
when you
idea.
feel like you've
Maybe
seen
you might-
everything rolling, we can come out.
You got it.
Okay.
Yeah, I see it.
I think you're given some great imagery at this distance.
Okay.
There's something there, so you
Oh,
see the
yeah, I
fins.
did hear that.
Somewhat?
The above that shrimp to the right.
So Tim was interested in
It's in
seeing
your
whether
shot.
some of the shrimp
It's
were
that little
gravid or
chimney
with
behind
eggs.
it.
Somewhat? Behind it? Sorry.
So,
Speak
So
up.
most of them are
It's like
crawling
a
not with their abdomens up.
monocle or
Am I
something.
correct?
So okay, so there's 2 places
I
that you
don't
can
see
see
it.
eggs on
Bottom
shrimp.
left
Sometimes
of the
depends
Sorry,
on
I'm
what
backing
stage
off.
they are at, I think. So they carry
Oh, more
them in
crabs. I hear you.
within under the
See
dorsal
that
surface of the
little
carapace sometimes, so you can
left
see
of the
them,
image.
see the eggs just through
No, I'm not
the
seeing
dorsal
it.
surface,
Sorry.
but then
Went off
also
screen.
they'll move them
All right.
at a
I'm
later
gonna keep
stage
going down.
I'll
down
just
to
keep my
on
eye
the
out.
underside
But
of the
it looked like
abdomen.
that monocle
and sort of
fins,
carry
maybe
the
potential.
last stage of the eggs, and then also
All right.
sometimes
Hold
juveniles.
on. You got it. Yeah.
So
You
we're
gotta
going to keep
almost-
an eye out for
you
that
gotta
while
almost
we're
yell
looking
at
at
me,
the shrimp.
Roland. I really can't hear
Huh.
what you're saying. You gotta
Okay.
like speak up
But
a
the
lot, Roland.
I'm not
Alvinaconcha
sure why.
gastropods, which are
think
not
just
the sort
clear
of flat ones we're seeing right now,
helps.
they're the much more rounded,
There. Now
typical
say it.
snail-like
Okay, how's that?
morphologies.
Yeah,
Those are
I
actually
turned them
hairy,
off so I could hear
which
you.
is
Ah,
just,
copy.
it's just really
So
interesting.
what were you asking
Where?
for?
Can you point that out for me?
So on the
So, you
left,
can't see any right
immediately
now.
Oh,
top
well, that would
left
be
of
why
the
I can't
image,
find
see the fuzzies on the rock
Oh, I do see that.
above the shrimp.
Yes.
All right, shore-based scientists. Look,
Okay.
there's like
Yeah.
a
They
moment
have
of
not
us not
seen
talking.
that
You should
yet,
jump in
and-
right now.
I
So
not
these
yet know
limpets are
what it
likely,
is.
sorry, I'm just going to use that
But
opportunity.
there appear to be
These
some type of
limpets are
worm
likely
monocle feelers
Shinkai
that
lepis,
open,
as
they're
Stace
opening
has
and
pointed out
closing.
before.
So
Yeah,
Thanks
okay.
very much,
if we
Stace.
get closer to something, there they on the bottom
I don't
left.
think we're,
Yeah,
I don't think we're
see.
sure
And dead
which
center right
species,
now,
as
too.
there are a
Oh,
couple
there's a couple
species
down
which
there.
occur around here.
So just in a second,
Oh, no.
we'll see that.
Maybe, yeah, no, there are a couple
Can
species
comment
which
on
occur
it.
around here.
Hey, those are barnacles.
Oh, yeah! Good spot. So we've
See
got
what happens
some,
when you let them talk
I
to you?
know. So Santiago, if you're watching, we do have some barnacles
gonna
on
hold
screen right
there.
now. You can see their cirri
Want to light it
extended.
up. I don't want to get too close.
And these are likely Neoveruca barnacles, possibly Neoveruca brachylepidodeformis.
Those are very cool.
So if anybody's tuned into the dives yesterday, I was very quiet, and the day before. I'm just going to make up for that today, tenfold.
It might be nice if we let some of them talk. You know. They're also experts in their fields, so it would be nice if we let some of them have a chat every so often.
Yeah,
just
that wasn't
a little
so bad, even without autos.
So as Deb said, feel free to chime in at any point, onshore scientists. I'm sure most of you are far more experienced than I am and have far more to say than I do. So please
like how quickly our
chime in
snap
at any point.
And actually, sorry, for
Zoom
Bill,
for 5 minutes
can
and
you
scan around. But
give me a hint of when we think we're about to see the flange that you'd like to try to drop the
kind
high
of,
T-probe at? Just let me know.
Delta difference did we do from the time we were zoomed in and dropped down?
We went from delta as in how far from the wall?
No, from top down.
Oh, we were
how
up
much
at
did
20.
we scan down?
We were up at 27, and now we're at 14.
This is the Space in the Woods hole. I think I only saw just a couple or few of the Alviniconca, which is the characteristic hairy male
for
here.
the zoom, you mean? How much did we change?
Oh,
maybe half a meter or one or something.
And
yeah,
my guess is that the hairy male will be down at the base of the chimney in a more cooler area.
If I had moved 15 meters that time, I would have been zooming by.
Great, Stace, and I think we're going to try to head down there a little bit more later in the dive.
Patty, I'm looking at
maybe a little tilt down on the main HD camera, look below us as we're headed in that direction.
Go ahead, Bridge.
Are those barnacles, or is that some other animal?
I believe those were the barnacles. Yeah, I think what you were looking at was the barnacles. We're a little bit
about the A-frame.
We're a little bit delayed. So
yeah,
we're
just
now looking
can I
back
confirm
at the main structure of the chimney?
It's about the A-frame of rotation. Center of rotation is about the A-frame.
All right, shore-based scientists. Diva went to go get some tea, so now is the time for you to talk.
Excellent. Yeah, we're comfortable with that.
So, Deb, I'm looking at the camera 2 view on Sirius, and it looks like maybe there's a flange below and to the left of where the ROV is.
Yep, that's what I see too. Do you want to try to sample there? Not sample, sorry. Do you want to try to deploy the high T-probe?
Yeah, unless you have something else you want to do.
No, I think that would be great. And that's what I'll relay that to the pilot and see if it's a possibility.
I suppose maybe we should see
Can we zoom the black thin smoker straight ahead?
We can do that as well.
Straight ahead or the one coming from the bottom of the screen?
Could you zoom in on both of them so you can zoom in on the little one first and then the bigger one?
Okay. Yeah, you can come in. Great. So, what's the word, watch lead? I heard you discussing and I couldn't.
Sorry, so what we'd like to do after imaging the close-ups of these little orifices. Oh, they're so cute. And what we want to do after this is, if you look at the Sirius view and you look to your left, so you're panning left, there looks like a flange. And what we want to try to do is deploy the high-temperature probe at that flange, sort of underneath the flange.
Okay.
If we see water, I mean, we want to make sure that we're seeing flow there before we do that.
Okay, so you want to poke underneath.
Yeah, and it should be more non-destructive than trying to get the high T probe into one of these little orifices. Generally quite difficult to aim the high T probe down into
Roland,
one of those
come
orifices.
out. I don't see the up and to the right one you're talking about.
Look at all that black smoke. Oh, this is great. Just great.
This is incredible.
Oh,
Love it. There's also a flange right in front of us, actually. Oh, yeah, a really big one. So what we want to do is we want to look at below these flanges and see if there's definitely fluid beneath them.
Yeah, culminating beneath it.
Right.
If we want, we can zoom in and then come down the smoke and let it be revealed.
So, Bill, what about this flange that's center right now?
Yeah, you can zoom in.
Oh, you won't see it anymore.
So to get full. Now
look at it, it's just absolutely billowing out of there.
And reveal it.
Okay. Wow, that crab's in a really weird shape. Position.
Yeah.
I would guess that one that was white would have a better chance of having fluid coming out around it.
All right.
One that is
Not white?
All of a sudden. All at once.
All right, we'll take a look. Oh, look at that nice orifice. Oh, I think that shrimp just got into a fight with a crab.
It's like giving small commands.
Because they're blind. Yeah, so because they're blind, they tend to bump into each other quite a lot. But also, the crabs do tend to be predatory or scavengers.
Are they predators of the shrimp? Or is that too
I'm not sure. Onshore scientists, do the crabs eat the shrimp?
For about 5 or 6
or do they tend
seconds.
to go for easier prey like the limpets or the gastropods?
Do they also eat
microbial
mat?
Yeah, or do they just eat bacterial mat?
Okay.
I'm thinking because they're predators or scavengers. Go on.
Thanks.
This is Patty from shore. What I've seen, especially with the round snails, is that when we have tried to sample them, the animals that come in to eat them are preferential.
Okay, so this is the sort of area you were thinking of prodding? Like that shelf?
Up and under.
Up and under this thing?
I think.
I thought they were sort of describing the white one to the left.
Okay, so pilots, what we were hearing, we may have to actually turn towards the left instead, because this one, we don't see as much of that white bacterial mat. We're not sure, although it looks like there's a black smoker under that flange. Look at that. Can you see that, Diva?
Yeah.
So,
it doesn't look like it's billowing as.
I don't know. Look at it off to the right. It's there. But I think what we were going to do is hope to aim for one of the ones that's whiter. So we want to look for one that's white but also has fluid underneath it. And we're going to ask the scientists on shore to help us with this.
Okay, I'm going to drop a little bit.
So scientists on shore, start looking.
All those chimneys in front of it, so it makes it a little harder.
Right here with all the shrimp?
Yeah.
Yeah?
So just keep, so shore-based scientists, just keep eyeballing, and somebody shout out when you see something that looks like it could be a good place to deploy the high-T probe. And if not, we'll keep moving.
I think it helps to get a little bit lower and then look up, so you can see the flange under part.
So the arm is 6 feet long.
I'll ask about that. So pilot?
Yes.
There's a suggestion that perhaps if we go down a little bit and look up, we'll be able to see how much fluid is flowing.
Okay. I can go down and look up. Can't probe from there very well.
No, it would be more of identifying where it is we'd like to probe, and then we'll come back to it.
So this is definitely where the vent sort of ends and we get into that brown stuff. You can see right here is the line. So I'm down and looking up.
Can we zoom in a little bit here?
Absolutely. There's definitely a flow. It's just how much is good. Come on in.
Something partial.
Thanks.
And then can you pan up just a little bit?
Panning up a touch.
So how does that look for everybody? Does that look like a reasonable place to try to deploy the high-T probe? Shore-based scientists.
Bill, it looks to us like that is black smoke getting trapped underneath a flange there.
Yeah, that looks like a good spot.
Okay.
It looks like there's water pools underneath it, huh?
Yeah, I agree. So just to make sure, I want to be crystal clear on this. So this is a good spot. We're going to try to deploy the high-T probe here.
Yeah, and that they'll want to poke the probe upwards, under. Make sure they understand what a flange is and that it's sort of an overhang that has, and there's water that's trapped under the overhang.
Yep. Okay, will do. So, pilots?
Yes.
So what we're going to want to do is deploy the high-T probe here if it's at all a possibility. I don't know if it is, but that's why I'm asking.
Yep. We're, co-pilot and I are going to take a look and situate ourselves and make sure that we are content with this location and that we can actually reach.
Perfect.
When all that is cleared, then we will go ahead and deploy.
Okay, and then the scientists wanted you to make sure, and I'm sure you already know this, that these flanges are areas where it's an overhang, and so you want to push the high-T probe sort of back into and below that probe, where there should be a pocket of hot fluid.
Okay. Yeah, I understand.
So I'm more concerned with where you're landing and on the
guys,
right side. Have we imaged all that? Made sure there's nothing coming up into us?
What we're going to try to do right now is we've done a lot of really great imagery
I'm not going to land. I'm
this
probably going to auto
30-meter tall
here.
chimney.
I'm just trying to get a good distance
We've taken
before
a look.
I auto.
We've sort of imaged around the top. We've zoomed in a lot.
I'd probably
seen shrimp.
back
We've seen limpets. We've seen crabs.
Think I'm too close forward?
We've seen barnacles. And what we're going to try to do now is deploy our high-temperature probe so that we can get a read for just how hot this chimpy is.
Arms around
and so
too a little.
Our pilots are sort of setting us up and putting us in the right
down,
position.
maybe.
And they'll let us know when they're ready to sample. Or sorry, to deploy. We're not sampling. We're deploying
Auto
the probe.
XY, auto depth.
Bring in your center.
No, we don't have any concerns on the wire.
Hold. Here. Come on,
Thank you, Bridge.
autos. Come on, autos, grab.
Pilots, bridge is going to change ship heading just a couple more degrees.
Auto is better up top than it is here, but...
Video, can you, Sirius is a little hot. Right. This is honest lifestyle. Thanks. Thank you. Are you full wide there?
Yeah.
Sirius is full wide. All cameras are full wide.
Okay. Want to spin us up?
Yeah, you're spun up. I'm just
going to reach out and see how close we actually are.
Let's tilt down. Let's see where your skids are.
Skids. Skids have lots of room. There's nothing but that black brown stuff here below.
I just want to make sure. Thanks.
Happy? Good?
Yeah.
All right. So. Yeah, I want to make sure everybody's comfortable before we reach.
So you are spun up. Let me give you a little bit more, actually. What would you like me to do with the wings first?
The wings are fine, I think.
Does that help at all? A little bit, maybe?
Yeah.
Let's do the same thing here.
Keep the autos. Okay.
I think we've lost the audio from the ship temporarily.
Are you sure? Did you kick the craft box off down below?
I didn't mean to.
Yeah, I'm pretty sure that's what's happened. It should be coming back shortly.
Did.
Chris and Bob, we can hear you perfectly. Can you hear us?
Do you know what switch it is?
Now we can.
Okay. Oh, there it goes.
So, just to put this in everybody's mind, we're at the 2-hour mark out of a 6-hour dive.
Got a little more light on you.
All right. Got good comms.
After we take this temperature, we should think about what else we want to do here, and
for the sake of
think about moving on.
Bill, I think that's a really great point.
Totally
so
still as we can.
Why don't we have that conversation? Obviously,
they're
we want
wandering
to deploy the probe here. Does anybody want to sample anything here? Do we know?
No, so what I think we should do is, and please others chime in, is that I think we should take the high temperature measurement and then on our way moving out of this area, go through the mid zone and the peripheral zone, which we will do on our way out of the area. And then
see how it goes here.
And then head on to the next side, because I think we can easily lose loads and loads of time here, and I don't think there's anything here that needs to be sampled just yet.
Okay. Shore-based scientists, does everyone agree that we don't want to sample here?
If anybody disagrees, please say so.
Mini Zeus is
Well, Bill, I'll just add one more
Can you bring that down a little?
Of course.
Guessing from the
Alright, yeah, you can draw, bring the drawer, port wing box inboard.
Which camera do you want on it, Mini Zeus or Titan?
I'll just get the Zeus on it for now.
Probably the biggest and most spectacular chimney we're likely to see. So, you know,
port wing coming in.
Yep.
It's certainly
great. That's good.
Of the dive, but I think it would also be valuable to see what else is at the sun site, even if it's smaller or whatever.
Bill, I think that's perfectly reasonable. I mean, certainly, as you said, we stayed here because this was an excellent example of a really large, nicely active, black smoker chimney. I completely agree. I think that we just want to make sure that because this is a community-driven science expedition, we just want to make sure that everyone agrees that it's time to move on and that we could think about sampling later after we've gotten a little further and maybe checked out some other sites. But we do want to make sure that everyone is on the same page.
I guess the main question is, does anybody have any sample?
You want to train Mini Zeus down onto where we're prodding so I can tell how far in we are?
Gotcha.
Thanks.
Come down.
Can we please have the temperature reading out on our camera number 3? Thank you.
Yes, I will ask.
All right, thanks.
I'll bring it up for you.
Super. What's going on here? Oh, the autos are flying away on us here.
They're walking a little.
Walking a lot. Still at lock. What's going on? Come on. Figure out that you don't know where you are. Come back to the wall, please.
Wand angled at about 45 degrees up to
That was
get
so
the
weird.
Underneath.
I wasn't touching anything.
Yeah.
Might just be. It might be like walking on flows.
I guess so.
Like on a.
That was really far away.
Just DVO, I mean.
Yeah. Okay. A little closer.
Okay, so on camera, on stream 3, you should be seeing the high temperature probe. Of course, we're not here, so I should probably be looking. There we go. Look at that. It's over here, Diva, just in case you're interested.
So, Deb, make sure they get the tip angled up so it's in the fluid under the flange.
Will do. So, pilots?
Yeah.
Not just the smoke that's going up towards the flange.
Go ahead.
So what you want to make sure you're doing is actually aiming the probe up. Like, so you want to point the tip of the probe.
I'm scratching the bottom of the rock right now. All the shrimp are mad and running away. So.
Well, if you were a shrimp, you'd probably run away too.
Yeah, so I think, it's definitely spiked. You can see it now. We're at 8 or 10 now or something.
Yeah,
you can't quite
10,
see it from across.
10 is probably not what we're looking for. We should...
I'll try to turn it up higher. Here.
Yeah, 140, 160.
There's 140.
You might be almost at the end of your...
Okay, so we're up at 140 right now.
Celsius.
Yes.
Yeah, 160 Celsius even, right? What are we at now? 200?
150 something.
4 degrees.
Oh, 150 something.
Oh, no, it went way up.
Yeah, Patty, you're a little bit delayed versus the regular video streams that are coming out. You might want to refresh your browser.
Okay.
I'm going to draw out
it's going
it
up
for
to 200.
Yeah.
It's passing 200.
Could try that. Go real slow. I don't want. Every time I move the arm, it's throwing the autos off a little bit.
Okay. I'm going to have to lose this for 1 second.
Yeah, let's keep it then. Let's just keep that up.
Or you can.
Yeah, I could draw her out. It's probably fine though.
Thanks, Josh.
Oh, it's 5 minutes into the next watch. I was wondering why all these people were here. I thought they were just excited. Yeah. Okay. Do we want to... I don't want to trade the watch like this necessarily.
225.
So, shore-based scientists, do you think we want to try to also deploy the high-T probe in an orifice now? Because we're not really hitting above 240 or so, or 235. Do we want to try for one of the other, for the black smoker orifices?
Yeah, I think it's basically where the temperature, where the fluid, is coming out clear before it becomes black, which we have seen quite a couple of on this chimney so far. I think there have been a couple of us talking about it. Jill McDermott, who's following along, who's worked at many hydrothermal vents, as well as Deb and I.
Deb, is there a camera that can see where the tip of the probe is? It's hard to see from camera one.
I don't know. That's a good question.
Do you want me to try to do any more wiggling around in here?
There's a better view from the mini Zeus as far as looking in, and we could tilt it up a little bit and zoom in.
I don't know that we're streaming the mini Zeus though.
No, we could, but we could.
Okay, I will ask video.
Is there any more crevices you want me to poke? Anything like that?
Hey, video.
Go ahead, Washley.
Can we stream the mini Zeus view on one of the cameras on one of the streams?
Or can you see is the probe tip up in the fluid under the flange?
Personally, I can't see because honestly, I'm a little far away from the cameras. But
can we tilt up the mini Zeus slightly and then zoom it in?
Would that help?
Video, where do we put it?
Really?
On stream 2. Okay. All right. So if you guys want to switch over to stream 2, if you're at home,
in the hole,
you can take a look at where it's a little different view of where the high T probe is.
Do you want a different crevice here?
Okay, that's a good view. Yeah, okay, looks like it's up in there.
And Copilot, we could suggest zooming mini Zeus a little bit.
To try
okay.
That, smoker that
I don't want to ask her to. But
go ahead, video.
I agree.
You can zoom mini Zeus.
Zoom mini.
Okay, that's what I'll ask them to do. Thanks so much, guys. You're doing an awesome job.
You can hold there.
Pilot?
Yes.
So I think what we're going to want to do is to try to deploy the high T probe in one of the orifices.
Okay.
In one of those black smoker orifices. Is that okay with you?
You know, I'm sure Josh will be very willing. He's about to take over the watch, but I'll let him answer that question.
Josh, what do you think? Oh, yeah. Okay. I think we're good.
All right. So, we're pretty excited about putting the temp probe in these, so I'm sure he's
all right,
stoked.
so
I'll pull this out.
Those of you that are watching or listening at home, we're about to have a little ROV watch shift change, and what we're going to do is redeploy the high temperature probe into one of the small black smoker orifices, and I'm going to tell you, this is actually often more challenging than it looks. You kind of look at it and say, no problem, we can totally deploy a probe there. But it's actually a skill as well as an art form. So we'll see what we can find when we get closer to one of the black smoker orifices.
Thank you.
For those of you who are just joining us on this dive, you have picked an awesome one to join on. So we are here at the Marianas Back Arc Spreading Center. We're at, I believe, right about 17 degrees north. Yes, we are. And we are at a depth of 3,285 meters. And we have discovered a 30-meter tall chimney that has never before been explored. There was a hint of this from an autonomous vehicle survey, which took pictures and did some chemical and particulate sensing. But we did not know we were going to expect a chimney quite this large. And so what we've been doing for about the last hour or so is imaging the chimney. We've been sort of looking up and down the length, sort of checking out how it formed, what it looks like, and what is living on it. And so we've seen many, many shrimp and limpets, gastropods of several varieties. We've seen some barnacles, and we just tried to we tried to deploy our high temperature probe here at a flange, which is a little part that sort of overhangs and sometimes captures fluid underneath it. And now we got to a temperature of, I believe, just around 250 degrees C. And now what we're going to try to do is actually deploy that same high temperature probe into one of the black smoker orifices. So it's where the black smoke is actually coming directly out of the chimney. And when we zoom in on it, one of the things that you will be able to see is that right at the base of those orifices, the fluid is clear. And that's the actual hydrothermal vent fluid. As soon as it comes in contact with seawater, it begins to precipitate minerals.
Hey, pilot. Hey, Nav.
And that's what turns those fluids into a cloud of black smoke.
So this little one up top, is that what we're talking about?
I think that one might be too little.
Too little? Okay. So maybe go up the chimney a little bit?
Yeah, let's see if I can. I need a different view, I think, in order to see this. Can we zoom out, or are we already zoomed out?
We're full wide on D2.
Okay. Do you remember where that other black smoke orifice was, Evo? Is it off to the right? Yeah. I think if you pan to the right, yeah, up and to the right, there were two orifices next to one another, and one was bigger than the other one. Hey, Jeff.
Way to the right in the camera 2 view.
All right, so pilot, Jeff told us that it's to the right and we might have to fly to get there. But he's going to help you now. Great.
Right. This nose sticking out, I came around on.
Yep.
You have to pirouette around the nose towards the right.
To the starboard. Okay. You want to keep an eye on my arm with the mini Zeus?
Yeah, I'll take it. We're nicely lined up here, so no worries about the tether.
That guy?
The squat lobsters
Those two
obviously
guys.
don't like it too
Or
hot.
that one. That guy. Okay.
There's no venting going on.
That should be easier to get into.
All right.
If we could snap zoom on them, just because we wanted to, it was a very specific orifice that had clear fluid immediately emanating out of it, but then went black, and that's going to be the hottest it can possibly be.
Come on in.
Is that coming out of a cone that sticks up? Oh, no, that's not.
I think you want to aim for that one that's the biggest.
I'm going to come around to the starboard here and take a look. Yep, go ahead.
They're so small.
Bit more zoom.
Okay, so pilot, did you copy that? We're going to go for this black one. Deb, are you happy with that? Yes. I mean, we don't see the clear fluid here, but
it doesn't matter.
As long as we get that probe down inside of it, it should be fine.
So you want to get the probe inside?
Yes, please.
Okay. I will come on out, video.
Oh, look at that. Did you see there was a crab sitting on top of that tiny little pinnacle?
No, I didn't.
It's very cute.
We'll zoom in again.
So shore-based scientists, actually, I've been, you've been doing a great job of telling us which feeds you want. Which
do you guys know, do we have a way to record temperature probes as samples?
We'll continue to make sure that you get what you need.
It goes into SCS, doesn't it?
To be able to explore this area.
Well, but do we officially say at this time we temp probe and it was 250 Fahrenheit? Or is anyone recording that?
That would be in the metadata somehow. It must be. I guess I don't know.
I guess that's a question for the back row. Watch lead, co-pilot.
Yes. Oops. Yes.
Just question on recording these max values. Is that part of your sampling GUI? Or are you?
Is it recording, you mean?
Well, we know the data is definitely recording somewhere, but I was wondering if there's a log of it or a.
I don't know. I thought you guys recorded it, so I don't know that we had it in a
Go ahead, Bridge.
I would note the time.
Yeah.
Note the time? Is that what you said?
Ballot direction.
Yeah. Okay.
Let's see. It looks like it was officially set to be south. Is that correct? Yeah, let me get back to you in a minute.
Yeah, and it's in the event log.
Yeah, that's a good point, Carl.
We can certainly search through all the data later, but it'd be good to.
Oh, yeah.
These are our first two ever, so.
Makes perfect sense.
Yeah.
I guess Nav can note it too for any confusion.
So it was 12:02 our time, basically, when we did the first flange measurement. Okay.
Think I'm close enough for this, Carl?
All right, so we're going to try to deploy the high temperature probe here at the surface.
A little closer, I think.
I'm just worried about this.
Yeah, I think right now it's going to vary a bit as we work around these structures. Right now, I think our bailout direction should be southwest.
You're worried about the ledge above the right skid?
You know,
yeah, just
obviously get more accurate than that, but it doesn't really matter. I mean, to
come up a little bit.
That would be fine. I think it would be good to be chatting about it fairly regularly as we